Seeds with 2 cotyledons are called dicotyledons, and seeds with 1 cotyledon are called monocotyledons. Mono means 1. Now the main function of the endosperm is nothing but to provide nutrition to the developing embryo. Apomixis is nothing but the production of seeds without fertilization, and this can be also called as parthenocarpy, meaning without fertilization the seeds develop. Hello everyone, a warm welcome to this concept class on biology for class 12th. I'm Dr. Divya, biology faculty. So this is the last session of this chapter wherein we'll be discussing some topics under sexual reproduction and flowering plants. So in this chapter, that is the last few topics, that is the post-fertilization structure, we'll discuss about the seeds, how the formation of the seeds occur without fertilization, and all that we will discuss under this. So talking about the seed, so seed is a fertilized structure or a fertilized ovule. We call it a fertilized ovule because if you remember the previous sessions, you would have known that the pollen grains which are released from the anthers, they come and fall on the stigma. Now, if the stigma and the pollen grain are compatible with each other, then the pollen grain, it releases its or it produces the pollen tube or the germ tube, and through that germ tube, the germ tube will reach the micropyle of the ovule. Now, the micropyle is the region where the opening is there, and towards the micropyle, there are cells which are called as synergids. And in the pollen grain, that is in the generative cell of the pollen grain, 2 male gametes will be there. So those 2 male gametes will be carried by the pollen tube and it will be delivered into the synergid. Now in the synergid, the pollen grains, one of the pollen grain will go and, now in the synergid, one of the male gamete will go and fuse with the egg cell, and the other male gamete that is there, it will go and fuse with the central cell. All this we have studied in the previous session. You can have a look at that. So now what happens is the egg cell will develop into the zygote. The zygote develops into the embryo, whereas the polar nuclei will develop into the endosperm. All that we have seen, or the primary endosperm nucleus and then the primary endosperm cell. Now all that we have seen that. So here, what happens here is, now the ovule that is there, it is the one that after fertilization, that is the zygote, will develop into the embryo. Now there the seed setting will start. So therefore seed is nothing but it occurs after the fertilization process. So therefore where inside the ovule. So therefore seed is nothing but a fertilized ovule. Now the seed, it is covered by hard seed coat. From where did the seed coat come? We had seen the structure of the ovule, right? Anatropous ovule, wherein I told you that in the anatropous ovule, it is covered by the outer integuments. That is, it is covered by the integuments, right? The outer integument and the inner integument. The outer integument which is not continuous and the inner integument which is continuous. So all that we have seen. So the integument of the ovule that is there, that will harden. It will become hard and it will form the seed coat. But still the micropyle is still present on the seed coat. Micropyle, a small pore. If you see the bean seed, very clearly, you can see just below the hilum or somewhere above or below the hilum, you can see a small pinhole there. So that is nothing but the micropyle. The micropyle is still there, which is clearly visible in the seed coat. So now we'll look into the basic structure of the seed. So in the seed, the outermost structure of the seed is the seed coat. From where did the seed coat develop? It is nothing but the ovules, the integuments of the ovules, they harden to become the seed coat. Next comes the radicle. Radicle is the region of the seed from where the root arises. So radicle region, it is the region where the root arises. Next comes the plumule. So plumule is the shoot tip. So for the region where the shoot arises, we call as the plumule, or the shoot tip is called as the plumule. And from that shoot tip, later on, the 2 leaves will come out. And therefore, the plantlet will be called as the 2-leaf stage. And next, the cotyledons. Cotyledons are nothing but the seed leaves. So you can see here 2 cotyledons. Therefore, this is a dicot. Apart from that, some of the dicotyledonous seed, they are endospermous, and the endosperm provides nutrition to the developing embryo and all that. And as you can see here, they are covered by a seed coat. And you can see from the plumule region, the first true leaves have arrived. So Plumule leads to shoot development. Radicle leads to the root development. So based on the number of cotyledons, the seeds are of two types. So it is dicotyledons, di meaning two. How many cotyledons? Seeds with two cotyledons are called dicotyledons and seeds with one cotyledon are called monocotyledons. Mono means one. So there are two types of seeds, dicot and monocot. So this is a dicot. All the leguminous seeds that are there, they are the peas, beans, the chickpea, the channa, or all the leguminous plants, they have the dicotyledonous seed. So these are the monocots. So monocots, they'll have only a single cotyledon. The best example that I can give you is the maize is a monocot. Next, talking about the dicotyledonous seed in detail. So dicotyledonous seed, again, it is covered by a seed coat. The outer covering of the seed is nothing but the seed coat and it consists of two layers. How many seed coats are there? Two. The outer layer, which is called as testa, it is very, very thick. So the outer layer is the testa. It is thick. And the inner layer, it is the tegma or tegmen, which is thin. So the seed coat consists of two layers, the outer testa and the inner tegmen. Testa is thick, tegmen is thin. So remember this testa, outer, tegmen is inner. Next, there is some scar in the seed. If you carefully observe the bean seed, you can see a white scar in that. That is nothing but called as the hilum. So this scar, if you can see here, this one, it is the hilum. This one, it is the hilum. Now this hilum is the scar. From where did the seed get that scar? Like how we have a belly button, right? So belly button is the scar where we had the placenta or the umbilical cord connected between the mother and the child, right? Same way here, the seeds, if you remember, I told you the ovules are connected to the ovary or walls of the ovary or to the tissues present in the ovary through placenta, which acts just like an umbilical cord delivering nutrition to the ovule, right? That placenta, when it falls off, once the seed develops, the placenta falls off. There a scar is left. So that scar is nothing but the hilum. Next, it has a small pore which is called as micropyle. Micropyle was already there in the ovule. If you remember, we had studied, right? It had two ends. One is the micropylar end and other one was the chalazal end. So micropylar end was the opening and it was through these opening that the gametes could enter and the fertilization process could take place. So it is a small pore. So you can see that micropyle or the small pore even in the seed, it will be very small. Either here or on this side you'll have the micropyle or the small pore. So this pore not only allows the entry of the gametes when the seeds are still in the ovule stage, but also the pore, it helps in the transport of oxygen and water into and out of the seed. So for exchange of gases and water also the micropyle helps. Next, talking about the cotyledons. The cotyledons are nothing but the seed leaves, wherein in dicot, two cotyledons will be present, and the cotyledons, they help in the storage of food. Therefore, they provide nourishment to the developing radicle and plumule. So, they are filled with lot of nutritive substances. So, whenever the radicle needs for developing roots and the plumule needs it for developing the shoot, nutrition is supplied by these cotyledons. Next comes the radicle. So, radicle is nothing but the embryonic root and the plumule is nothing but the embryonic shoot. So, in the seed, there is an embryonal axis wherein the top end that is there. So, this is the radicle which gives rise to the root and the plumule portion which gives rise to the shoot. Where they lie in the embryonal axis. So, this is the structure of the dicotyledonous seed. Now, let's look into the monocotyledonous seed. This along with the diagram is very, very important. The monocotyledonous seed, usually they'll ask about this in the exam with a neat label diagram. Same like how dicotyledonous seed had the seed coat, same way monocotyledonous all seeds also have the seed coat, but the only thing is in dicotyledonous seed, the seed coat, they had an outer layer called testa and inner layer called tegmen. But in the case of monocotyledons, the seed coat are fused with the fleshy part of the monocotyledon, which is nothing but the pericarp. So this is the outermost layer, which is the seed coat. It is fused with the pericarp. Next, inner to the seed coat, they have endosperm. So the major portion, this large portion which is there, the bulky portion, that is called as the endosperm. Now the main function of the endosperm is nothing but to provide nutrition to the developing embryo. So the endosperm, it lies near the small embryo and it is large and it is starchy. Because it is starchy, it has a lot of starch in it which provides nutrition to the developing embryo. Next, there is one more important layer in the monocotyledonous seed. It is called as the alluron layer. So this entire layer that is there, it's the alluron layer. This alluron layer is very, very rich in protein. So that is why cereals and pulses and all that are quite rich in protein. So they are very rich in protein and they are a special proteinaceous tissue that surrounds the endosperm. So you can find them only in the endosperm. So this is, next we have the cotyledon. Now how many cotyledons are there in the monocot? There is only one cotyledon. That is why it is a monocot. It has a single cotyledon and the cotyledon is termed as scutellum. So this is a single cotyledon of the monocot. It is scutellum. Then it has the plumule and the radicle. Plumule is the region where the shoot arises and radicle is the region where the root arises. And the plumule is protected by a protective covering which is called as coleoptile. Coleoptile. And the radicle is also protected by a protective covering called as coleorhiza. Coleorhiza. So this is about the monocotyledonous seed. And apart from that, in the previous slide itself, I told you they are covered by seed coat and they have the endosperm. And then they have this layer which is the alluron layer. Alluron layer. So this is the structure of the monocotyledonous seed which is very important in examination point of view. So next let's study about why are seeds important. So seeds are extremely useful in agriculture because if we need, of course we have now other forms of vegetative propagation and all that wherein the stem and all that will be cut and the new plants will be generated out of it. Or we have tissue culture wherein some tissues of the plant material will be taken, it will be put in a nutrient media and optimum conditions will be provided for that. And there later on calyces or mass of cells will develop and that from each of the cell we can develop a new plantlet. Still though all that is there, the traditional method of agriculture is the one that is of great use because it is cost effective as well. So that is why. So seeds are extremely useful in agriculture. So what are the other important or what are some of the very, very important applications of seeds we will see in this. So seeds, they help plant species to be colonized in different areas because seeds, they can be easily carried away by birds. So birds, they take the fruits, they fly over to another area, they eat the fruit there and they drop the seed. Now that seed will germinate there. So a new species of plant can be introduced in a new area because of the seeds. And seeds, they provide nourishment to the young seedlings. Because in the seeds, they have the cotyledons. So, the cotyledons are the one that actually provides the nourishment to the developing seedlings. And the seeds actually ensure protection to the young embryo. Of course, the cotyledons, they take care of the young embryo apart from providing nourishment. And seeds are the ones that results in variation with new genetic combination because seeds can be set in a flowering plant because of cross-pollination also. Now, because of the cross-pollination, so when new set of traits combine, what happens? A new plantlet that develops will have some new characteristics which can be useful for agriculture. So, therefore, it results in variation with new genetic combination and it can be easily stored for future use. So, in airtight containers or in plastic bags, we can neatly store the dry seeds and whenever needed, it can be put to the soil and germinated or sprouted and then germinated. And they have long-term viability. So that is why seeds, it is easy to store, because seeds, they can be dried to a moisture level of just 13%, and then they can be stored for long time, and again they can be sowed and we can get new plantlets out of it. So therefore, these are some of the important applications of seeds. So based on the presence or absence of seeds, now we'll look into the two categories of seeds. So two types of seeds we had studied, that is monocotyledon and dicotyledon. Monocots can also have endosperm, dicots can also have endosperm, some of the species, but based on whether they have endosperm or whether they do not have endosperm, seeds are classified as non-albuminous or ex-albuminous seeds and albuminous seeds. So non-albuminous or ex-albuminous, meaning they do not have endosperm. They lack endosperm at the maturity. Of course, endosperm will be present when the seeds are very, very young. That is, before they have germinated, endosperm will be present because they have to provide nourishment to the developing embryo. No, so, but when they germinate and mature, at that time the endosperm will be lost from that seed. So, example, pea, bean. Next, albuminous seeds. Seeds which have endosperm are called as albuminous seeds. Example, wheat, rice, etc. So, usually the dicotyledonous seeds, they lose their endosperm during maturity, but the monocotyledonous seeds such as wheat and rice, they have their endosperm throughout. So, based on the presence or absence of endosperm, the seeds are of two types, non-albuminous and albuminous. So, here you can see a large quantity of endosperm in monocotyledonous seed. So, this taken the example of maize. Whereas in leguminous plants or dicotyledonous seed, endosperms will not be there during maturity. Moving further, let's study about the small term that is given in the textbook, which is apomixis. So, what is apomixis? This is a question. Define apomixis or explain about mechanism of apomixis. So, apomixis is nothing but the production of seeds without fertilization, and this can be also called as parthenocarpy, meaning without fertilization the seeds develop. This mechanism actually produces clones. Now, how they produce clones? So, say for example, if seeds are developed by fertilization, there are chances that most of the characters from the male plant can come and incorporate there, right? So, we have a plant A and plant B. Now, plant A is the female, plant B is the male. So, from the male, whatever gametes are being delivered into the female, the male's character also can get into the progeny, right? It's just an example to give you, like us. Do we look exactly like our parents? No, but we have some of the traits of our parents as a collection, right? So, that chances are there. Now, if a clone of me or you is produced, they will be exactly like you or me, right? So, that is what is called as apomixis in the case of plants. So, in the case of plants, apomixis is nothing but as and how the parent plant, whatever character the parent plant has, the same character the progeny will have. So, it is a clone of one another, right? No changes at all. So, that is why such why such clones develop, because of seeds developing without fertilization. So, without fertilization means obviously chances of other characters getting mixed up is not there. So, therefore, apomixis is nothing but the setting up of seeds without fertilization, and this mechanism usually produces clones, and therefore it is asexual reproduction, because fertilization is not at all involved here. Male gametes are not at all involved here. Therefore, it is called as asexual reproduction. And apomixis can occur in different ways. So, one thing is, now the question that arises is, then how did the seed set without fertilization? Something from the male gamete should come and fuse with the female gamete if the fertilization has to occur and the seeds need to set, right? But here now you're telling us that there is no fertilization happening, then how did the seed sets? So, what happens here is, in the ovule there will be some tissues which are called as nucellus. So, these nucellar cells which are diploid, they are usually present just outside the embryo sac. So, if you remember the structure of the ovule that we had done in the previous sessions, you can see that they were covered by integuments. Inner to that was the covered by integuments. Then we found the embryo sac. Now around the embryo sac, they have a patches of cells which are present. Those cells are called as nucella cells, and the tissue is called as nucellus. Now those nucella cells, they start to divide. Continuously they keep on dividing, and when they continuously divide, they enter into the embryo sac. First, where they were present outside the embryo sac. Now because they're continuously dividing, there is no space for them inside there in that region outside the embryo sac. So what they do is they start entering into the embryo sac, and later those nucella cells which entered into the embryo sac, they develop into embryos. Now embryo formation occurred, and therefore in this way multiple embryos will get collected inside that, because whatever nucellus or nucella cells entered into the embryo sac, they all will develop into embryo, forming polyembryonic condition. Poly means many embryos will get formed, and this will later on develop into the seed. So seed setting will take place. This is usually seen in citrus, mango, etc. So you can see polyembryonic condition. Can you see here one embryo, the second embryo, the third one? So you can see multiple embryos being formed there. So that is polyembryonic. Now here fertilization itself did not take place, but didn't the fruit develop? Didn't the seed happen? Yes. So setting up of seeds without fertilization, this is called as seedless. Okay, but seedless doesn't mean that seed is not at all there, but it is just that fertilization would have not occurred. So that is about apomixis. So sometimes the egg cell that is there is not formed as a result of reduction division. We had studied, right? That is, one of the male gamete will go and fuse with the egg cell to form the embryo. Now their fusion was required, but in the case of apomixis, what will happen? Fusion is not required. The nucella cells that were there, they themselves will enter into the endosperm, and they themselves will get converted into an embryo, forming multiple embryos. That is polyembryonic condition. And apomixis is extremely useful these days because they produce clones. Clonal production, reproduction is possible, and new hybrids can be produced in lesser time, because all those embryos we can remove and we can take it to the tissue culture laboratory and develop our own hybrids, that is with the desired characters that we want in that plant. We can do it, and disease-free plants can be produced. Why? Because here fertilization is only not happening. So how will the disease from another plant come to that particular plant, right? Not possible. So therefore disease-free seeds we can get, or disease-free plants can be produced, and it is very cost effective. Why? Because polyembryony, multiple embryos we will get, therefore multiple plantlets we can develop out of it. So these are the uses of apomixis. So next, moving to one more term called as dormancy. Dormancy means going into dormant. The seed will go into dormancy, saying that no, I am not going to germinate. You provide me good conditions, good water, good temperature, good nutrients and all that, then only I will germinate. That is called as dormancy. So that period until when the seeds do not germinate, that is called as the dormancy period. So usually the seeds that we store in our house, the beans and all that, do they germinate on their own? No. Right? Only when we put it in the water, then they germinate, because they are getting enough water there. The amount of the good temperature that is needed for them is also available. So therefore germinate. So therefore dormancy is called as the inactive stage of the embryo, is called as dormancy. So dormancy usually is attained by the seeds during unfavorable conditions. Say for example, some drought is there. So during that drought time, if the seed germinates and it develops into a plantlet, will it get enough water for its survival? No, the plant will eventually die, right? So that is when the seeds undergo a mechanism called dormancy, wherein they will not at all germinate, and once the rainy season starts, no, that time they will germinate. So that is how the dormancy gets break there. So what are the events that occur during dormancy? We will see. First is hardening of the seed coat. Now, of course, the seed coat consists of two layers. It is the testa and the tegmen. The testa is the thicker, harder one. The tegmen is the thinner one. But the seed coat will become even more harder, so that the seed cannot germinate, because harder the seed coat, germination is more difficult. So during dormancy, the seed coats will become hard, and also the micropyle will become very, very minute, so that the proper movement of water and oxygen, or water and gases exchange will not take place, so that the seeds will not germinate. So that is the two things that happens during seed dormancy. So how the seeds undergo dormancy? First is they will make their seed coats very, very hard, so that they don't germinate, and also that small micropyle, or that small pinhole opening that was there, that will become even more smaller, so that the proper exchange of gases and the transport of water into the seed will not take place, so that the seeds don't germinate. So therefore the dormancy can be broken. Now the seeds, once enough favorable condition is available, the seeds will sense it, and then they will automatically germinate. So that is how the seeds break their dormancy. So these are the two important events that occur in the seed during dormancy. Next, we'll talk about the fruits. So, seeds are nothing but the fertilized ovules. Now, fruits are nothing but the ovary. So, fruits are of three types: true fruits, false fruits, and parthenocarpic fruits. True fruits, they develop only from the ovary. So, the cells of the ovary, they develop into the fruit. False fruits develop from the thalamus as well as the ovary. So what is the thalamus? It is nothing but the receptacle, right? So thalamus is nothing but, so this we can say, this portion, it's the thalamus here. This is the sepal or the calyx. Now this entire thing that you see, it's nothing but the flower, the female reproductive part that develop into the fruit. So this is the sepal, the thalamus. So some of the fruits, thalamus is nothing but it's also called as the receptacle. We had studied in the first session of this chapter. You can have a look into it. So some of the fruits, they develop from the thalamus as well as the ovary. We call it as the false fruits. So apple and strawberry are the best example for false fruits, apple and strawberry, because they develop not only from the ovary but also from the thalamus. Next is parthenocarpic fruits. These are the fruits which develop without fertilization, meaning they are seedless fruit. How they happen? We had studied in apomixis, right? So these are the three types of fruits. Now talking about the structure of the fruit. So the fruit is covered by pericarp, and it is nothing but the wall of the ovary. So the wall of the ovary that is there, that will develop into the outer layer of the fruit, which is called as the pericarp. And inner to the pericarp, or sometimes the pericarp, it may be fleshy. Say for example in guava, mango, etc. The fleshy part that we eat, that is called as pericarp. It can be fleshy also. Sometimes the pericarp can be fleshy. Or it might be dry, like in mustard, walnuts, all that it is dry. The pericarp is dry. So in mustard, walnuts and all that, the pericarp is dry. So this is about the fruit, nothing much. So in the fruit, we have the seed, already the seed structure we have already studied about the seed. Now the seed is covered by the pericarp in some of them, or the fleshy part of the fruit, it is covered by the fleshy part of the fruit. In some of them, the pericarp can be fleshy, in some it can be dry. So this is about the fruit. So this was about the session wherein we learnt about the post-fertilization structure. We learnt about the dicotyledonous seed, the monocotyledonous seed, and also learnt about how seeds develop without fertilization. And also we got to know about the structure of the fruit and different types of fruits as well. So this was about the session, the last session of this chapter. So I hope you found this session useful. So we shall meet again in the coming session wherein we'll take up new topic and new chapters and discuss it. So see you in the coming sessions. Thank you.